topic on biotechnological interventions for fungal disease management in oil yielding crops. As Dr. Shivang has already introduced me, I am Dr. Prashant Yadav, scientist biotechnology from Indian Institute of Day-3 Mustard Research, Bharatpur, Rajasthan. And as far as our topic concerns, there are 9 major oilseed crops in India, among which 7 are considered edible oilseed crops and 2 are for industrial purposes, are grown in our country. And among these, the 3 oilseed crops, that is rapeseed, mustard, and soybean and groundnut, are occupying the major area, almost more than 85% contribution in national edible oil basket. And among these 3 crops, the rapeseed mustard, which is grown in Rabi season, and the major growing states are Rajasthan, UP, Haryana, MP, and Punjab. And I am mainly talking about these rapeseed mustard crops only because I am working on these crops. So coming to the future challenges in edible oilseed crops. So as we all know, with growing population and depleting natural resources, it is becoming very challenging to feed our growing population. And it is projected that around 10 million people will be there by the 2050 year. So with increasing food demand, we have to increase our oilseed production as well. And when oilseed crops are concerned, there is one more interesting phenomena is added, that is per capita consumption. So due to the changing dietary habits, the per capita consumption has skyrocketed. It was around 14 kg per year per person in 2011, but after only a matter of 10 and 11 years, it became 19.7 kg per person per year. And it is expected to grow around 24 kg per person per year by the year 2030. So population is also increasing, as well as our per capita consumption is increasing. So it is becoming very challenging for the oilseed crops to fulfill the demand of the growing population. And we all know that the India only produces around 40 to 45% of its domestic demand, and around 55% of the edible oilseed in India we have to import. And last year we have imported around 16.5 million tons of edible oils. And besides this, due to the certain environmental challenges are also there, like climate change. We all know that frequent drought, floods, and extreme temperatures are affecting our crops and reducing its production, as well as declining natural resources like land degradation is there, soil erosion is there. We have to use novel fertilizers. So limitation of water is also. And with all these changing climatic conditions, we have seen that pests and diseases are also becoming more severe. Some of the minor pests, now it become major pests. So with the changing climatic conditions, we also have the challenge of pests and diseases. And today's topic is also on fungal diseases in oilseed crops. Besides all these things, one important parameter is also there, that is nutrition quality. Because last year global hunger index, it is shown that in the figure that around 35% of Indian children below the age of 5 are stunted, and it is now becoming very important not only to secure the food security but as well as the national nutritional security. And oilseed crops are major contribution in the nutritional security along with pulses crops. So coming on the rapeseed mustard crops, it is not a single crop; it is a group of crops mainly derived from Brassica rapa, Brassica nippus, Jungia, and Brassica nigra, and one more crop which is Taramira called Iluca sativa. So these crops are mainly grouped into 2 groups: rapeseed and mustard. So the basic difference, the basic phenological difference is there. The leaf, when attached with the stem with the petiole, it is called mustard. When there is no petiole is there, then it is called rapeseed. Besides these, certain other parameters are also there, like normal mustard crops are having more pungent flavor and also used in condiments. But rapeseed crops are, especially particularly Brassica nippus, has very mild taste. And it is due to the low glucosinolate and low erucic acid content in Brassica nippus. But Brassica rapa, there are 3 subspecies of Brassica rapa, that is Brassica rapa yellow sarso, Brassica rapa brown sarso, and Brassica rapa toria. All these varieties has good pungency as compared to as mustard. So Today we are mainly focusing on the Indian mustard, that is Brassica gentiana crop. And coming to the trends of area production and productivity of rapeseed mustard crop in our country, you can see that the area as well as production and also the yield of the rapeseed mustard crops is increasing with day by day. However, our total edible oil production and our total edible oil demand, which is we have to import, is not decreasing. So we have to focus on the increasing rapeseed mustard production in our country. And there are certain challenges in our country to which hinders the optimal yield potential of the rapeseed mustard crops. So coming to these challenges, there are mainly two types of challenges: biotic stresses as well as abiotic stresses. So coming to the biotic stresses, the major fungal diseases in rapeseed mustard crops are stem rot, white rust, alternaria blight, and powdery mildew. Some of the person who has joined from Rajasthan, that they can they maybe know this phenomena that the stem rot is now becoming major challenge in Rajasthan state, and it is caused by the fungal pathogen Sclerotinia sclerotiorum. And this is a polyphagous fungus, and it is not coming only to rapeseed mustard crops, but many vegetables as well as pulse crops. So it is polyphagous and very notorious fungus. It germinate and attach on the stem, and since it is a necrotropic fungal pathogen, it is means make a lesion and decay the plant stem, and all the aerial plants then become dead, and seeds were shrinking as well as very low oil content and nutrition value is very declined. So it is causing major havoc in Rajasthan. And coming to the virus, it is caused by the fungal pathogen Albugo candida. It is a oomaxid fungal pathogen, and this is also becoming very challenge in almost all the mustard growing states, especially in Rajasthan, UP, and as well as in Haryana. So, white rust actually causes the white pustules on the lower side of the leaf, and this decreases the photosynthesis efficiency of the leaf, thus means lowering the sink source relationship. This pathogen also causes stag head formation in the inflorescence of the mustard plant, and thus reducing the yield. Coming to the alternaria blight is also a major challenge, not particularly in the state of Rajasthan, but the Tarai region of UP as well as Bihar has major severity of this pathogen. And alternaria blight is caused by Alternaria brassicae, mainly this pathogen, and this is also a necrotropic pathogen. So there are two types of pathogens: necrotrophs and biotrophs. So necrotroph pathogens, those fungal pathogens which mode of survival is to first kill the plant with the toxins and then survive on the dead tissue. This biology is called necrotrophs. The biotrophs actually thrives on the living host. They didn't kill the plant completely, but hijack the plant's system to give nutrition to themselves and advance their progeny. And now coming to the fourth pathogen, powdery mildew. Actually, powdery mildew was not earlier, it was not a major pathogen in Rajasthan, but now it is becoming a major pathogen because of the changing climatic condition. So powdery mildew is now also very much a big problem. So I will talk about one of my PhD topic, which is proteomic study of alternative blight resistant rapeseed mustard. So, as I earlier tell that Alternaria blight is caused by Alternaria brassicae pathogen, it is causing the concentric ring. And you figure in, you can see that the concentric rings are there, the light pink color as well as the brown dark color. Their concentric rings are there. Within these rings, you can the enlarged images are there, and finally, on your left bottom side, you can see that the Alternaria spores are visible in these concentric screens rings. So for our study, first for any kind of study, first you have to isolate the pathogen because no contamination should be there. The single pathogen should be there. So for that we have to go for single spore culture, and then we have multiplied the culture and means obtain the multi pathogen spores. And these pathogens were spores then again sprayed on three different Brassica species from Brassicaceae family. These are the group from Brassica sino species. One is Brassica tinctia, our Indian mustard, and other two varieties are its wild relatives, that is Cyanipes alba as well as Camna sativa. Cyanipes alba has very light seeded, very light yellow coated seed, and also called white mustard, and grown in very small pockets in India for condiment purpose. But Camellia sativa is very wild in India, and in Canadian countries it is have some industrial purposes. But in India, we mainly grow Brassica junctia. So these three species we have taken because the scientific alpha shows some of the medium level of resistance, but Camellia sativa shows high level of immunity against this Ehrlichia brassica pathogen, and we want to understand what is the pathogenicity behind this, and we go for the proteomic route to study this host pathogen interaction system. So the earlier slide which I have shown that we have identified the single spore culture, and these spores can inoculated plant leaves, and you can see that after 12 days of post inoculation, Brassica junctia leaves showing the big lesion size, always secondary infections. But in Synapis alba, only primary infection is there, but Camellia sativa, no infection or disease progression is there. So we can say that the Brassica junctia is highly soluble for Alternaria brassicae, and Synapis sativa has moderate resistance against Alternaria brassicae, but Camellia sativa shows high degree of resistance against this pathogen. So we have did the pipette blue staining to see for the necrotic tissues in the leaves as well as silics on these Brassica species plants, and we found that the Brassica junctia showed high level of necrotic tissues in leaves as well as their siliqua, and Synapis alba shows some of the necrotropic spot in their leaves, but no such sign was observed on the siliqua. So the seeds are safe from the infection of this pathogen in Synapis alba. In Camellia sativa, no necrotic spots were seen in leaves as well as in silics. So again, the dab staining, this is a kind of staining which usually we do to identify the amount of reactive oxygen species produced in the plant tissue. So in leaf tissue, you can see that Brassica junctia after infection produces a lot of reactive oxygen species, whereas Synapis alba have relatively low but still high ROS species in the leaf tissue. But Camellia sativa, however, not produces much ROS. Actually, necrotropic pathogen, the ROS is helpful for biotropic pathogen, but for necrotropic pathogen, ROS actually kills the plant. So the plant tissue, then the dead tissue is food for the pathogen. So it is pathogen will grow means easily on the dead tissue. So we found that in Camellia sativa, the ROS scavenging system is very high. So that is why it is one of the reasons that the Camellia sativa shows high level of resistance for this pathogen. Coming to the proteomic study, we have took three different time intervals. We found that the 12-hour and 24-hour post inoculation samples giving more optimum results. And you can see that the control and infected 2D gels actually have some of the spots are showing differential expression, and we identify those spots and analyze in PDQuest software. It is a digital software which analyzes the digital image, and identify the spots having differential expression. So here in this figure, you can see that the right graph, which shows that the blue line, the spots points above the blue lines are having more than twofold expression upregulation, and the spots having below the red line showing the twofold decrease in their expression in control as well as the infected 2D gels. So these spots are taken from the gels, and then trypsin digestion were done, and then we have to go for the mass spectrometry, that is multi-top MSMS analysis. And by this analysis, we got the peptide mass fingerprint, and that peptide mass fingerprint will scan throughout the available protein databases and found some of the very important proteins involved in the host-pathogen interaction, like Werke protein, cysteine-specific methyltransferase, and all these proteins. So here are some of the spots which we have highlighted. You can show that some of the spots are overexpressing, and as well as some of the spots are reducing in their expression in control as well as infected protein samples of these three species. And finally, we some of the genes, for some of these genes, we have conducted the real-time PCR study to validate these results. Found that these results are actually correlated, highly correlated with our proteomic special studies. So this is the kind of studies which we actually we have done in our PhD, and we found some of the important genes. In future we will target those genes for means some of the down-regulated genes. Suppose a particular protein is down-regulated and cause resistance. Then through CRISPR-Cas or gene editing, we can highlight those genes, mutate those genes, and get the desired plants. So this is my PhD research work, but future work will be done for getting the resistant plants for Alternaria brassicae. Now coming to the second disease, that is powdery mildew. Here again we have done this experiment on these three Brassica species, that is Brassica junctia, Cyanipes alba, and Camellia sativa. And again, the similar kind of study we have done, the histopathological study. We identified the spore from powdery mildew infected leaves from the field, and then these spore culture when sprayed on the these three species. And here we found that the Cyanipes alba is showing much more resistant for this powdery mildew pathogen, but Cyanipes Camellia sativa is showing highly resistant. So the same plant showing highly resistant, the Camellia sativa showing highly resistant for Alternaria brassicae, that is a necrotrophic pathogen, now becomes susceptible to the biotrophic pathogen powdery mildew. So here I think its mechanism, the less production of ROS is helpful for the necrotrophic pathogen, but it is not good for biotrophic pathogen, because in general scenario, the biotrophic pathogen, when plant recognize the invasion of biotrophic pathogen, then it produces ROS and kill its tissue surrounding the plant spore or plant hyphae. And then plants, since biotrophic plants live on the living plant, then if the surrounded dead tissue will actually kill the pathogen also. So we have identified the Cyanipes alba for this powdery mildew resistance. Here presently I am working on developing white resistant Indian mustard, and there are two loci already have been mapped for white resistant, that is ACB1 A4.1 and ACB1 A5.1. And these two markers, actually we have a genotype called DRMR MJB Pathis 35. In this genotype, we have both these two alleles. So we have took this genotype for pyramiding in one of our elite mustard cultivar, that is Giriraj, and we have developed a backcross population. Through backcrossing, we are actually developing the nails. Means Giriraj should have only these two alleles from DRMR MJB 35, and rest of the genome should be from Giriraj. So here we are doing the foreground selection for MJB Pathis, and we after going through the BC6 F1 generation, we will then again do the background selection for Giriraj genome recovery. And when the more than 97-98% genome of Giriraj has been recovered, and only these two loci have been incorporated, at that condition we will get the nil for Giriraj having these two registers. So why we are incorporating two alleles? In biotrophic pathogen, the R gene concept is there. So single gene can have resistance for a particular biotrophic pathogen, but it give high resistance, but simultaneously the resistance can be break very soon, because pathogen has to evolve against for only single R gene. But when you are incorporating more than two R gene, then it is very pathogen for pathogen it is very difficult to getting mutation for all these three genes simultaneously. So for durable resistance, we have to go for the gene pyramiding. So this is study in advanced end, and the lower right figure, you can see that some of the lines having durable resistance, these two alleles actually showing greater level of resistance for this white rust. Now coming to the Sclerotinia stem rot, again there is not much resistance source available in Brassica gene pool for the stem rot pathogen. And since it is a polyphagous pathogen, it is a robust virulence mechanism. So what we have done, we have studied, screened our entire germplasm algae advanced breeding lines, and we found one line, that is DRMR 12/28, having high level of tolerance. We cannot say resistant, but high levels of tolerance, because we measure in terms of lesion size. So susceptible plants, the lesion size is more than 5 centimeter, we call it susceptible, and the lesion size with less than 3 centimeter after inoculation, then we call it as a tolerant, highly tolerant. So the 12228 has showing high tolerance, as well as some of the mutant lines are showing tolerance, but it is in not very advanced state. However, we are developing real population with this tolerant pathogen to actually try to map the QTL responsible for the stem rot tolerance. And we project we also is looking for metatranscriptomics studies to identify the effector molecules responsible for the pathogenesis and help the stem rot pathogen to invade the brassica plant. So by identifying the particular important effector molecules, then we can go for genomic. We can use genomic tools to develop resistance for this pathogen. So we are also doing this. So with this, I would like to conclude my talk, and the future prospects should be using CRISPR-Cas9 or prime editing for precision breeding. So this can, because genome is a very powerful tool, and we can mutate the target gene, and this is a very less time consuming process. And since regulation is also very liberal in case of SDN1 as well as SDN2 techniques of this CRISPR-Cas, so we can use this technique to develop fungal disease resistance plants. And one more, speed breeding is also very important. Nowadays it is becoming very important because it lessen our generation advancement time. And within 1 year, we can theoretically we can grow 4 to 5 generations of this. So developing mapping population as well as double haploid population or any kind of population as well as transgenic population is become very easy, and the time will be very less. Besides this, we have to go for the multiple omics techniques for deep understanding of host pathogen interaction, and by understanding this system, we can then develop resistant plants for these fungal pathogens. One more concept is there, pangenomics, because pangenomics is like combining the entire more than one genome to get the maximum allelic diversity in the genome. So suppose all the resistant lines, breeding lines genomes as well as their synonymous genomes, when we combine these genomes, we get the full range of allelic diversity with this. And by studying this pangenome, we can then select what are the best allelic combination responsible for resistance against these fungal pathogen and use this. And then again, AI and big data analysis is also very much important because when we are using go for multi-omics techniques, so omics is particularly in any technique when the sample size is very large, data is very large, then it is called omic technique. So large set of data should be there, and the phenotypic data is also very much important. So the large number of phenotypic data as well as the genomic data, then we have to go for the AI as well as machine learning tools to easily identify and develop these pipelines to identify the resistant genes for these fungal pathogens. With this, I would like to conclude my talk. Thank you very much to all for patience hearing.